Hello students, we are into a new chapter, chapter 4, plasma membrane. You have already studied about cells. You know that cell is the basic unit of life. You have studied about the different parts of the cell and their specific duties. Now it is very interesting to know that some substances pass through the cell. So this is a wonderful mechanism that the cell can carry out. The cell is covered by a thin membrane called the plasma membrane. This is a covering. It provides a covering to the cell and it acts as a barrier. It keeps the cell constituents in and it keeps the unwanted substances out. So to understand further this mechanism, let us see what substances should go into the cell and what are the substances that should come out of the cell. Oxygen, glucose, proteins, fats, vitamins, minerals, these are all nutrients essential for the cell and therefore these will be transported into the cell. Whereas carbon dioxide and other wastes should go out of the cell. What is essential to the cell growth will go inside and what is not essential will come out of the cell. So transport is one of the main function of the plasma membrane. To understand this function, we have to do various activities. So let's get started. Preparing solutions. Let us first prepare a sugar solution. So for this we need sugar and water. So if you dissolve sugar in some amount of water, the sugar will completely dissolve and it will form a sugar solution. Now how do we prepare a saturated solution? Saturated solution is a solution in which no more solute can be added. So to prepare this kind of a solution, you will take 100 ml of water in a beaker and add sugar or you can also add salt. Stir till it is dissolved and next add again some amount of sugar and repeat till no more sugar is dissolved. Like when you add excess, it remains at the bottom. So this means no more sugar can dissolve. Then this solution is called a saturated solution. Let us now observe three different solutions. Take 3 beakers and pour 100 ml of water in each beaker. To the first beaker, add half teaspoon of sugar. To the second, add 1 teaspoon of sugar. And to the third, add 1.5 teaspoon of sugar. Now we will see that the solution in the third beaker will be the more sugary one. What is the reason? It has higher concentration of the solute. So this beaker, if it has to become more dilute, we must be adding water, that is to the third beaker, to make it like the solution of the first beaker. Let us do an activity of observation of material in different solutions. For this we require 2 beakers, tap water, sugar and dry grapes. For the procedure, we will take 100 ml of water in a beaker and we will keep a dry raisin or a dry grape in it. Leave it aside for 1 hour and then observe what has happened. Take it out and compare it with a normal dry raisin. What do you see? Is there any change in the size of the raisin? Yes, after getting soaked in water, it will definitely become swollen. You may try this with some dried vegetables or wilted vegetables also. Then in another beaker, take 100 ml of saturated sugar solution. Now the dry raisin which has become swollen is taken and dipped into the second beaker which has saturated sugar solution. We will leave this overnight and observe the next morning. What will be the change in size of the dry raisin? So in the first step, when the dry raisin was put in water, water moved into the raisin and the raisin became swollen. This swollen raisin, when it is kept in saturated sugar solution, again becomes shriveled or shrinked because water has moved out of the raisin. How does this happen? The cells in the outer layer of the raisin have permitted water to pass through. The process by which water has moved into the resin and out of the resin is called osmosis. Osmosis is the movement of solvent molecules from a region of low concentration to a region of high concentration across a semi-permeable membrane. Let us do another activity to demonstrate osmosis. For this, we will require fresh, large sized potato tuber, glucose solution, beaker, water, a blade, petri dish, and a bell pin needle to mark the sugar level inside the potato cup. Peel the potato and slice the potato tuber into two equal halves. Scoop out the inside of the potato so that it looks like a cup. A tiny cavity is formed in a square shape at the center, and the base should have minimum thickness. Now fill up half of the cavity with freshly prepared sugar solution. Into the cavity, fix a pin in such a way that the mark is in line with the layer of the glucose solution. Set up this potato cup in a beaker that is filled with water in such a way that 75% of the potato is immersed in water. Leave this setup uninterrupted for up to 1 hour. Now notice the sugar solution in the potato towards the end of 1 hour. Now we have one more half of the potato remaining with us. We will use this potato cup also. So in this potato cup, we will take water inside the cup and we will take sugar solution in the beaker. The same experiment is repeated, and after 1 hour we will observe for the levels inside the cup and outside the cup of the potato. We will observe that the first cup which had sugar solution inside, the level of the sugar solution will rise inside the cup. Whereas on the outside, we will see the water level decreasing. An increase in the level of glucose solution is observed because of the entrance of water due to osmosis from the beaker. Now let us understand filtration. This again we have an activity and we require two beakers here, one funnel, filter paper, a retort stand, sugar, dye and wheat or rice flour, 500 ml plastic bottle and a cotton cloth. Arrange the filtering apparatus as shown. Prepare wheat or rice powder solution in a beaker by adding 1 teaspoon of powder to 100 ml of water. Then add 1 drop of iodine to the solution. Now pour the solution into the funnel. What will remain on the filter paper? We will observe that the flour or the rice flour powder will not pass through the filter paper and it stays on the filter paper. So these particles are large and they cannot pass through the filter paper pores. So cells also act in the same way while allowing substances to pass through the cell membrane. So now we can understand that plasma membrane can allow water to pass through and it can allow certain materials dissolved in water to pass through, but it will not allow large molecules and certain materials to pass through. So allowing only certain materials to pass through is called selective permeability. What are the functions of the plasma membrane? The plasma membrane provides a definite shape to the semi-fluid contents of the cell. It functions as a mechanical barrier that protects the internal contents of the cell. The membrane will determine what substances are allowed to enter and what substances are allowed to exit. The membrane can carry out endocytosis. This is because of the flexibility of the membrane, which enables the cell to engulf food and other substances from the external environment. This process is called endocytosis. It can be seen in amoeba. How amoeba acquires its food is by endocytosis. The cell membrane also helps in recognition. That is, it has substances on its surface which function as recognition sites and points of attachment. So these molecules which help in recognition are called receptors. Then there is flow of information. The plasma membrane allows flow of information amongst different cells of the same organism. That is, it allows for cell to cell interaction. Plasma membrane can carry out osmosis as we have seen in the experiments, and then cell continuity. Plasma membrane helps in adjacent cells to join and to form plasmodesmata and cell junctions. Then there is specialization. Plasma membrane gets modified sometimes to perform different functions. Example, absorption in the villi. Transport is one of the major functions of the plasma membrane. The membrane acts as physical barrier between the cell and the surrounding environment. The cell organelles also are bound by a membrane. So this membrane here will form a barrier between the organelles and the cytoplasm. The membrane can be said to be impermeable if substances do not pass through the membrane. It is said to be permeable if substances, both solute and solvent, can pass readily through the membrane. It can be said to be semi-permeable if it is permeable to solvent but prevents passage of solutes. And plasma membrane is supposed to be selectively permeable if the membrane allows the passage of some selected solutes and solvent. Now we have one more experiment to understand the movement of water in animals. So for this activity, we will require 3 beakers, a petri dish, salt, dilute HCl, cleaning acid, and 2 equal sized raw eggs and a cloth to wipe, a strip of paper for measurement, and 1 tablespoon. In the first step, keep the raw eggs in dilute HCl or cleaning acid for 4 to 5 hours. What happens? Eggshells get dissolved. Now you can take the egg out with the help of a tablespoon. Wash the eggs under tap water. Now these eggs will be de-shelled eggs, and measure the circumference of each egg with a long strip of paper and put a mark on the paper with pen or pencil. Then take 2 beakers. Prepare a concentrated salt solution in 1 beaker and place 1 egg in the concentrated salt solution. Take another beaker with tap water and place 1 egg in the tap water. Leave both the beakers aside for 2 to 4 hours. Now take the eggs out and wipe them and measure the circumference with the same strip of paper which you have already used previously to mark the circumference. What will we observe? The egg placed in salt water will shrink. Shrinking is because the water from the egg has moved outside. The egg placed in tap water will swell, and the swelling is because the water has moved inside. The process by which water molecules leave the cell is called exosmosis, and the process by which water molecules enter the cell is called endosmosis. Let us see how we can prepare a semi-permeable membrane. When you take a boiled egg and when you break it, you might have observed a thin layer under the eggshell. So this layer is a semi-permeable membrane. It prevents free entry of materials into the egg. Only certain materials are allowed to enter. You can isolate this membrane. Take 1 raw egg and keep the raw egg in dilute HCl for 4 to 5 hours. What will happen now? The eggshell, which is made up of calcium carbonate, will get dissolved. Now you can take the eggs out after 4 to 5 hours and then pierce a pencil-sized hole in the egg membrane. The shell will be removed. You will have only the membrane and it can be pierced. So all the contents inside the egg will be drained out. Only the membrane will remain. You can take the membrane, wash and use it for your further experiments. Now this is called a semi-permeable membrane. Now using the semi-permeable egg membrane, we can do an experiment. Take two egg membranes, three beakers, sugar, water, thread, measuring jar and a disposable syringe. Take one egg membrane and fill it with 10 ml of saturated sugar solution using a syringe. Tie the mouth of this membrane bag with a thread. And then this bag is now placed in a beaker which has 100 ml of tap water. Now keep the membrane in the fresh water beaker and leave it overnight. Take the second egg membrane and fill it with 10 ml of tap water and tie it with a thread and place this bag in 100 ml of saturated sugar solution and leave it overnight. What will we observe? The bag with sugar solution would have become swollen because water will move inside, and the bag with tap water will shrink because water would have moved outside. What is the importance of osmosis to living organisms? Let us see a few points. Water enters the roots through osmosis. Water moves between the cells through osmosis. It helps in opening and closing of stomata. Osmosis brings about movement of water and minerals to certain plants. In our body, waste materials are filtered by the process of osmosis. Useful materials are absorbed along with water through osmosis. Did you know? 3/4 of the earth is covered with oceans. There is plenty of water around, but we cannot use it because it is salty. Only when we can remove the salts from it, we can use it. So Thomas Jefferson in the year 1791 invented a process called desalination. This is a process by which salt is removed from seawater using a semi-permeable membrane. So here salt water is separated from fresh water using a semi-permeable membrane. Heavy pressure is applied on the salt solution. So then what happens is water moves from the salt solution into the fresh water, leaving the salt behind. Now this process is known as reverse osmosis. Nowadays, domestically, this process is being used to filter water and to make it potable. Reverse osmosis is a purification process that uses a semi-permeable membrane to remove ions, unwanted molecules, and large particles from drinking water. It is a process by which solvent passes through a porous membrane in the direction opposite to that for natural osmosis when subjected to hydrostatic pressure greater than the atmospheric pressure. There are other ways in which materials move in a medium. Let us study one such process by following an activity. Go to one corner of a room in your house and open a perfume bottle in the corner of the room. What will you observe? You will see that the perfume smell has spread to the entire room. How did this happen? The smell has spread in the entire room uniformly, and even if you are blindfolded, you will be able to reach the scent bottle because you know where the smell is coming from. So, what is this process called? This is because of diffusion. There are certain other examples also for diffusion. For example, if you open a soda bottle, the bubbles will become flat after some time. This is a kind of diffusion in liquid. Another example is a drop of food color in a glass of water. So that food color will spread to the entire glass of water. So these are some examples of diffusion. Let us do an experiment with coffee powder. Take half a bowl of water and prepare a small ball of coffee powder. Put it slowly into the water and observe what has happened. You will see that the coffee powder has started dissolving in the water. You can do this alternatively by Pouring hot water on the pinch of coffee powder. So the color of the coffee powder spreads into the entire water. Another activity can be done with a small crystal of potassium permanganate kept in the center of a petri dish with the help of forceps. Carefully fill the petri dish with water using a dropper. Now you will observe that the pink color in the petri dish is diffusing into the water from the KMNO4 crystal. So the color spreads from the center to the periphery. You can repeat this experiment with other substances like a crystal of copper sulfate. The process by which these materials are spreading equally throughout is called diffusion. So, diffusion is the process by which materials move from a region of higher concentration to a region of lower concentration. Thomas Graham, a Scottish physical chemist, worked on the diffusion of gases. This scientist he measured the rate of diffusion of gases and also studied the diffusion of different substances in solutions. He concluded that a more soluble substance in a medium will dissolve faster than a less soluble substance. So, this is popularly known as Graham's law of diffusion.